So just like we did for glycolysis, I'm going to do a little walkthrough here of the steps of the citric acid cycle. So there's 8 steps in the citric acid cycle, all occurring in the mitochondria. And I'm actually going to start with what is technically step 0 of the citric acid cycle, or we called it step 11 from glycolysis, which is the pyruvate dehydrogenase chemistry. So pyruvate, 3-carbon alpha-keto acid. So we are going to decarboxylate that and take the 2 carbons and put them as an acetyl group on CoA. So that's the starting point for the citric acid cycle, is acetyl-CoA, and then that other carbon gets liberated as carbon dioxide. So we spend, here's that carbon dioxide that comes off. We spent a good bit of time going through the details of how that kind of process works, the 5 steps of the pyruvate dehydrogenase multi-enzyme complex with the E1, E2, E3 long arms, all of that. So we'll save that for another video to kind of walk through. But here's the details. Again, pyruvate dehydrogenase doing oxidative decarboxylation. So we decarboxylate, and it's an oxidation step. So we must couple that with a reduction, and here that reduction is NAD plus to NADH, right? It's always really important that we make sure that we understand when we're looking at species that are related by redox, understanding who is the oxidized form and who is the reduced form. Okay, so what's different with the citric acid cycle versus glycolysis is it's a cyclic process. So there's not really a starting and an ending. And that'll come into play a little bit too once we start talking about regulation of the citric acid cycle. It's not like you've got the start and the end when you're trying to figure out where does it make sense to regulate. So one of the things I'll highlight as we're looking through this image here is that some of these carbons are labeled, and I'll get to what those labels mean in a minute, but it's just saying, hey, if I know that this carbon is specifically this one, I can track it and follow it as I go through. So that's one of the important things I want you to know is which carbons come from where. So the start of the citric acid cycle is citrate synthase. We're going to take 4 carbons from oxaloacetate and 2 carbons from acetyl-CoA, and we're going to hook those together to make the 6-carbon citrate. So we'll go through the details enzymatically of that step, but basically these 2 carbons from acetyl-CoA are going to combine with these 4 carbons from oxaloacetate. I want to show you a little bit for how it happens. I'm going to kind of highlight that these 4 carbon atoms here, if you can imagine bending this guy a little bit in half, that's these 4 carbons. So these are the carbons from acetyl-CoA. You can see that label that we have here that reminds us of this. I'm going to walk through a little bit just to remind you how that happens. In the enzyme active site of citrate synthase, we are actually going to make this into a nucleophile. We're going to deprotonate it. We're going to have nucleophilic attack here. And because this is a carbonyl containing compound that we are not going to have a good leaving group, we end up protonating it to make that alcohol. So that's how this chemistry happens. So these 4 carbon atoms are these 4 carbon atoms. These 2 are those 2, and this guy is the nucleophile. So this is the new bond that we make right there. Okay, so that's one of those mechanisms we'll go through, but just to kind of highlight and show it there. So now we have citrate. I want to highlight just for a minute with citrate. Again, these were the 4 carbons that came from oxaloacetate. These were the 2 carbons that came from acetyl-CoA. This is the carboxylic acid that comes off first as our first CO2, and then this is the carbon that's going to come off as our second carbon dioxide. So one of the important things I want you to see is that even though the net is, hey, 2 carbons came in as acetyl-CoA, I lose 2 CO2s. If we put labels on these 2 carbon atoms, they're the ones that are up here. They're not the 2 carbons that come off in this round of the citric acid cycle as carbon dioxide. So that's a really important thing I want you to kind of be able to follow and trace. It's kind of a skill for learning the citric acid cycle, is kind of following and labeling carbon atoms. So it's not until these carbon atoms go through the merry-go-round, the second cycle, that they actually get eliminated. Okay, so here's citrate. Next step is called Econotase. So some of these enzymes have weird names because they were discovered before there was a formalism for how we identify enzyme names. So Econotase here is going to do some isomerization chemistry. Essentially, we're going to move this OH group from this carbon onto that carbon. So we've got citrate here, and then we've got an isomer of it. We've got isocitrate. So that's the first step, but let's talk about why that's important. We talked about when we were labeling these species, this is sort of a hidden beta keto acid. So here's our carboxylic acid that's going to be coming off as a carbon dioxide molecule. Here's the alpha carbon and the beta carbon. Now we know if that was a beta keto acid, it would decarboxylate easily. But it's not a beta keto acid, it's a beta hydroxy acid. But if we do oxidation chemistry, we've created that beta keto acid that can then decarboxylate. And that's what's going to happen here. So our next step, isocitrate dehydrogenase, does oxidative decarboxylation. So two things kind of happening at once. The first thing that we do is we're going to oxidize this alcohol group to a ketone. That happens because we generate a reduced NADH. Now, we don't have this structure that's shown. It's an intermediate in the enzyme active site that's not isolated, but what we really have is we have a beta keto acid. That easily decarboxylates, we lose CO2, and then we're left with 5 carbons left, and this is alpha-ketoglutarate. Okay, so I'm going to zoom out here just so we can see these next to one another. So this is the carbon atom right here that we lost our CO2 off of. And because we made it into a beta keto acid, it readily decarboxylated. Okay, so one more step here. Fourth step, we are into the last half of stage 1, which was chemistry on the substrate. So we're going to see the generation of our second molecule of CO2, and we're going to see a second NADH that's created. Remember, for your fundamentals quiz, I don't need you to know stoichiometry, but come time for the test, you are going to need to know how many of each thing are made. So this is the second place that we're going to make NADH in our citric acid cycle. So this is alpha-ketoglutarate dehydrogenase, and we're not going to study this enzyme too much because it is a homolog to pyruvate dehydrogenase. It basically does the same chemistry, requires TPP to decarboxylate an alpha keto acid. So we're going to see the same kind of chemistry, 5 steps, E1, E2, E3 arms, moving all over the place. But basically, we're going to be eliminating this CO2 group as carbon dioxide. So you can see the label there reminds us that that's the guy that comes off as CO2. The rest of this, just like we saw with pyruvate dehydrogenase, gets attached to CoA, so we generate succinyl-CoA. Okay, so that represents the completion of the first 4 steps of the citric acid cycle that generate all of our carbon dioxide. So 2 carbon dioxides came off, and now we've got a 4 carbon piece that we say sucks with its functional groups, because right here we've got alkanes. So the next chemistry is going to be reconverting one of these groups into a ketone, but we also have one extra little space, one step before this, that we can actually use this high energy succinyl-CoA intermediate to generate a GTP by what equates to substrate level phosphorylation. So let's look at that step. So the name for this enzyme is succinyl-CoA synthetase. I know it's a weird name. It's not synthase, it's synthetase. So it's important to remember that name, succinyl-CoA synthetase. Okay? And what we're going to do is essentially save the energy of that high energy thioester bond and use it to couple GTP and inorganic phosphate together to make GTP. So this equates to mechanistically and energetically a substrate level phosphorylation step. Now we've got succinate. And here we highlighted that succinate really sucks having functional groups because there's only these alkane 2 carbon atoms in the middle. If we just look up here right now where we need to get to, we need to have with oxaloacetate one of those methylene groups, so CH2 groups, become a ketone. So 3 steps to do that. And this is going to be a pattern we're going to see. So I want you to see it now and kind of get it in your brain, because this is a theme that we're going to see repeatedly when we do other sorts of chemistries, either working in the forward or the reverse direction. We're going to take an alkane, oxidize it to an alkene. We're going to hydrate an alkene into an alcohol, and then we're going to oxidize an alcohol to a carbonyl-containing functional group, in this case, a ketone. Okay, so we're going to see that same type of chemistry happen when we do metabolism of fats, because remember what fats are with fatty acids. It's alkane, right? So we're going to see that same chemistry there. And actually when we go to, that's oxidizing fats, when we go to build fats, we're actually going to be working in the reverse for that. So this is going to be something we're going to see again. So I want to make sure we have it up here, but let's go through the enzymes and the steps for that. So one of the things I want to notice, you see how this guy here, we knew when we had succinyl-CoA that carbon with that label was specifically up there. Now, how this gets a little bit different is this molecule has no stereochemistry with it. It's symmetrical, right? So that's why we see this 1/2 here, is we are essentially mixing. Either one of those carbons could be the one that has our label on it, so to speak. All right, so succinate is going to be oxidized to an alkene, which is fumarate. Fumarate is going to be hydrated to malate, and then malate is going to be oxidized to oxaloacetate. So we want to have that story in our head so that when I'm asking you those detailed questions, you can kind of piece together those steps. Okay, so succinate gets made into fumarate. Okay, the enzyme for that is succinate dehydrogenase. Now, dehydrogenase enzymes, anytime you see dehydrogenase, you want to be thinking that that is redox chemistry. But specifically, if we go alkane to alkene, we are going to be seeing FADH2 as the reduced cofactor that's made. We'll talk about this once we get there, but just to give you a highlight and a preview, succinate dehydrogenase is actually one of the enzymes in the electron transport chain. If you've studied the electron transport chain before, you know that there's complexes 1, 2, 3, and 4. This is actually complex 2 of the electron transport chain. So saying it here so that when we say it again later, hopefully it refreshes your memory. Okay, so now we've got fumarate. Okay, we do have some stereochemistry here. It actually is a trans-alkene there, so that is intentional. So fumarate, we can hydrate alkenes. So hydrating an alkene, and this is done stereospecifically in our enzyme active site. So we're going to generate L-malate. So we have a very specific stereoisomer of malate that we generate. Enzyme for that is fumarate, so here's where we see water. That reminds me, I got to go back to the beginning again and highlight there is another place that I want you to know where we have water, because we need 2 water molecules. So now we've got malate. Malate as an alcohol is going to be oxidized to a ketone. Anytime we see redox chemistry between oxygen-containing functional groups like alcohols and ketones, or carbonyl-containing functional groups, that's when we're going to see NAD+ and NADH. Again, you'll notice that this is a dehydrogenase chemistry, so it's telling us that we're doing redox. But in this case, we're oxidizing that alcohol to our ketone, and we have oxaloacetate, another alpha keto acid. So just highlighting here, here's the other place where we need water. So part of why I do these walkthroughs is to show you all the different places where we're going to see certain reagents or cofactors or things like that. So when we make this, okay, when we do this coupling chemistry, remember I showed you that this is going to be a nucleophile. It's going to nucleophilically attack this, and we're going to then protonate this guy to make our alcohol there. Well, at this point, it's still actually attached to our CoA. So the reason we need water here is we are going to hydrolyze this ester. So this essentially equates to a good leaving group. We're going to have this actually come in, nucleophilically attack. We're going to have a great leaving group here, and that's how we're going to get our CoA off. So we'll have another video here to walk through those mechanisms so you can see all those different pieces. All right, that's it for the 8 steps, well 9 if you consider pyruvate dehydrogenase, of the citric acid cycle.